Well, I appreciate being invited to meet with all you nice people and talk a little bit about my interest in conservation and how I deal with some of the issues that seem to be causing big problems nowadays. I started my work as an evolutionary biologist who was interested in amphibians, and I picked a group of organisms that has some of the largest numbers of extinct species that are known as far as a class of vertebrates goes. So it was hard to avoid conservation. I tried to avoid conservation because it's a whole lot easier to work with common species. You don't have to worry about permits or people yelling at you, and you usually have free access to people's properties. But when you start working with endangered species or threatened species, there's a bit of a problem. I mean, it does take you a lot of lead time to get all the permits in place nowadays, and I find it kind of hard. I've also had to change my research. It used to be that I would slaughter the animals in order to get tissues for my isozyme electrophoresis that I was doing population genetics on. Fortunately, today, all we need to do is take a little tissue sample, and we can extract DNA, and we can get all the information that I used to get with isozymes, and actually a bit more. And it looks to be in the future, we'll be able to use water samples to find out if an animal is in a pond or not. And that's kind of scary. I kind of like to get into a pond with my boots on and actually see the animals in order to find out if they're there or not. But that's what's coming. What's also coming, which is kind of scary too, is that we can do the whole genome of many individuals in a pond. We can actually tell, does this individual have genes which would enable him to survive in a different environment? I mean, these are so-called genes of adaptation. And this is on the horizon and not too distant horizon, where we can actually look at particular genes that enable animals to survive. And again, amphibians are a wonderful group of organisms because they're so-called R species. In other words, they have an intrinsic rate of growth. Their populations can expand amazingly. Therefore, they have to undergo great and harsh natural selection. For instance, each individual female can produce 300-400 offspring. If none of the offspring died, we'd be up to our armpits in salamanders. So obviously, there's been a lot of predation going on, and there's a balance in the community. The energy of these little larvae that are produced is transferred from the pond onto the forest environment, and there's a lot of species that rely on each other. So it's the whole biomass is very important. Now, habitat is the, am I too loud here? Okay. Habitat is a big problem, and all the legislation that has to deal with habitat. We have to protect the habitat of threatened and endangered species, and this is written into the Species at Risk Act. It's written into the Niagara Escarpment Plan. It's into the Greenbelt Plan. It's written into the Oak Ridges Moraine Plan. And if we destroy the habitat of a threatened and endangered species, then that's a big no-no. We can't do that. Now, we're hearing today that, well, maybe we can. Maybe we can destroy the habitat as long as we don't decrease the benefit of that particular area for these animals. But I just wonder how much of that is based on good science. I have a few pictures here. Oh, there's one, okay. For the past 30 years or so, I've been working with the Jefferson Salamander. And it's a very interesting salamander because it is involved with a genetic complex that I really don't have time to talk about. But it is rare and it is endangered in Ontario. Most of the populations have very few salamanders, and it's the males that are more endangered than the females as far as we can tell. There's not too many places left where this salamander is actually doing very well. There's some areas in Pennsylvania where it's not doing too badly, but even in Pennsylvania, it's now considered to be threatened. In the next picture, if if I can have there. This is a so-called vernal pond. Now, this is where these animals breed. At this time of year, in fact, tonight, these animals will be going down to these ponds, and they'll be breeding. And usually the males precede the females, but you can go to these ponds and see them this time of year. Now, normally these salamanders are considered mole salamanders. They're underneath the ground for most of the year, so therefore you don't normally see them. If you want to see these salamanders, you go out tonight to a pond. And then next week you won't be able to see them again until next March or April. So this is also a problem. Now, since you only see them in this pond, people say, this is the habitat for the Jefferson salamander, a vernal pond. In order to protect this habitat, we should protect a buffer around this pond. And let's make that buffer 30 meters to give them lots of space. And so this is habitat protection. 30 meters around a vernal pond will protect the Jefferson salamander. Now, this is not true. If we look at so we start to do some ecological work and doing hypothesis-based research. If 30 meters around the pond is the home of the Jefferson salamander, we should be able to find them in that area. So if we look at the next slide, here we can go into that pond this time of year, and we now use minnow traps, and we can sample the pond, see if there are any Jefferson salamanders in that pond. And in some ponds, in good areas, we have maybe 80 or 90 salamanders in one of these minnow traps. And they live together with yellow-spotted salamander, and they live together with this unisexual complex, which again, it's very complex. I don't want to get into it. Okay, the next picture. In order to figure out where the habitat is for these salamanders, what we first started doing was to use drift fences. And what a drift fence is it is a line of fencing that is parallel to the edge of the pond, and the salamanders are caught as they come to the pond to breed. And they go along the fence trying to find a hole, and they drop into a pitfall. And then we can go and look in the can traps, and we can find out where the salamanders are. Now, if we put these drift fences at different distances from the pond, we can see where the salamanders actually come, how far they come into the pond. We can also use the drift fences to look at salamanders when they transform, leaving the pond as they go into the woods. So we can, and these are wonderful. These are wonderful things to use. But this isn't quite good enough. We don't know exactly what they do. Where are they? We know that they came from here to here. We can take them here and put them over the fence and find they get the next fence and so on. We can also mark the salamanders, and we find that the salamanders every year come from the same places and go to the pond. And when they leave, they go in the same direction. So this is so-called pond fidelity, that the salamanders utilize the same breeding pond year after year after year. They come and enter from about the same points of the pond. If you have the next slide, please. This is just to show what the can trap looks like. And normally we monitor them pretty much all night and in the morning, so you can't leave them too long. And we have problems with rodents sometimes biting them, and big insects, and raccoons, skunks. So we usually hide them underneath a rock or something, so only salamanders can get in there. In the next slide, we found, actually a friend of mine who is a former student of mine has developed these transmitters, which are small enough that we can actually insert them into a salamander. These are radio transmitters, and we can follow the salamanders around. So this, it looks like a pretty big transmitter to fit in that little salamander. And we don't do it until we do it to females that have laid eggs, so that they have a big empty cavity inside their body cavity, and we can put the transmitters in. And it doesn't seem to hurt the salamander. Like, we sew them up. Salamanders are amazing as far as they don't get infected easily. And then we sew them up. And we even found that, unfortunately, the transmitters only last for about 3 months, so we can't follow them through the whole season. And but we have found salamanders with transmitters going back the next spring and lay eggs. So we know it doesn't really do a lot of damage to the salamanders if they do it correctly. So this salamander's looking at it kind of suspiciously. So we can have the next. So using these little radio transmitters, what we do is follow the salamanders around during the spring and summer and find out where they go. And so, and this is one. The big problem here is you can't put transmitters in every salamander, and it takes 2 students here their whole time, all summer, to locate about 14 salamanders. So we do, and we assume that what's happening in the few salamanders, the relatively few salamanders that we can insert radio transmitters in, we assume that that happens to all salamanders. And so our frequencies are based not on huge numbers, but on smaller numbers. And we find the salamanders move around. They move mostly when it rains. When it rains, they move more than it doesn't rain. Sometimes they sit in the same spot for a long time. They live in rodent burrows underneath the ground, underneath stones, and they wander a little bit, but not a whole lot. And we find that most of the salamanders go about 300 meters from the pond. So about 70% of the salamanders don't go much further than 300 meters. Again, it's based on small sample size. But some salamanders go up to 1 kilometer away from the breeding pond. So what do we think about our 30-meter buffer around a vernal pool as being a habitat for these salamanders? So we can now establish a habitat as being much more than 30 meters. The habitat would be habitat which would allow these salamanders to survive and do pretty well, be closer to 300 meters, and they can go about 1 kilometer. So if we draw a kilometer circle around a breeding pond, that's a considerable amount of territory which should be protected as a habitat of these salamanders. Of course, using these things, we also know that there are certain habitats that aren't frequented, that there are so-called critical habitat and so-called non-critical or non-used habitat. But we know, for instance, that, well, let's take an example. Here we have a forested area that was clear-cut some time ago. And now we have a field, say a soybean field, between the pond and the forest where the salamanders spend most of their time. They still go across that soy field to get to the pond because the salamanders can live up to 30 years. And they have pond fidelity. They use the same path back and forth to the pond. And so we're just beginning to learn what salamanders actually do. Unfortunately, we can't talk salamander. That would be really cool if we could. But by using some of these research techniques, we can get a pretty good hint on what's happening in the salamander population. I don't think I have any more slides to. Oh, yeah, there's sometimes to actually find the salamander, you can follow the salamander and get close enough to use the little probe, and when you can actually dig him out of a rodent burrow, so you can prove he's there. So all I'm trying to do is to say that we don't know very much about habitat for some threatened or endangered species. And I think it's fairly critical in order to make decisions that we understand a great deal more, especially if it requires destroying some of that habitat and assuming that the individuals would go to a new habitat. For instance, the chimney swift. That was a good example. If the same chimney swifts could abandon one chimney and go to a new chimney, that would be wonderful. But I would like to know that they are the same chimney swifts that go to the new one and not some other individual. So this is a kind of research question that should be asked in order to figure these things out. So I don't think I have anything more to say.
Thank you, Dr. Bogart.